Hello and welcome to the Biochemical Society's Portland Press. I'm pleased to welcome you to this webinar, which is part of the Biochemistry Focus ECR webinar series. The series features research from different areas in the molecular biosciences, as well as practical sessions to support career development. The Early Career Research Series aims to present topics of interest to and research from the early career community. I'm Amy Lewis, your chair for today's session. I'm a postdoctoral researcher at the Blizzard Institute and a member of the Biochemical Society's Early Career Advisory Panel. In today's ECR webinar, we will be hearing from 4 early career researchers working in the field of signalling. Before I introduce our first speaker, please note that questions will be asked after all 4 presentations at the end of the webinar, but you can send in your questions at any time. Type it in the question box as shown in the image on the screen, stating who your question is for. We will try to answer as many as possible at the end. Our first speaker today is Dr. Andrew Holding from the University of York, and his talk, Next Generation Approaches to Understand Tissue-Specific Regulation of the Glucocorticoid Response. Dr. Andrew Holding is an early career researcher at the University of York, leading a team that focuses on steroid hormone receptor signaling in breast cancer. Andrew initially trained as an organic chemist, receiving an MChem from the University of Oxford and a PhD from the University of Cambridge. In 2009, he was awarded an early career development fellowship at the MRC Laboratory of Molecular Biology, where he focused on elucidating the structure and function of proteins-bound complexes using mass spectrometry-based proteomics. From 2013 to 2019, he led the experimental research lab as part of the Markovets group at CRUK, expanding his experience to include genomics and computational biology, and was awarded the Turing Fellowship in 2018. Alongside his research, Andrew has a successful career in science communication, producing radio and written content and working with organisations such as the BBC and The Guardian. He won the inaugural CRUK Rising Star in Research Engagement Prize and was named one of the 175 Faces of Chemistry by the Royal Society of Chemistry. Today, Andrew will talk about his research into steroid hormone receptor signalling in breast cancer and related disease. Over to you, Andrew.
Well, thank you for that introduction, and thank you for inviting me to speak today. So my talk is going to be on what I have started to do, well, will be starting to do here in York. I'm very lucky that this work has been funded by the BBSRC this year, so I'm excited to talk about it. But to lead in where and how we got to this position, I'm going to have to talk a bit about what I've been doing for the last few years. So despite the title saying glucocorticoid receptor, I'm going to spend quite a bit of time talking about the estrogen receptor. So the reason I've been interested in the estrogen receptor and steroid hormone receptors in general is due to this slide. So this is from the US government, and what it's showing is when we talk about breast cancer, in terms of clinical treatment, we can subdivide it into multiple different types, and the most common form is this labeled here is hormone receptor positive, HER2 negative. So the hormone receptor, the estrogen receptor, drives the growth and the proliferation of the tumor. And that's 73% of patients, so this is a huge number. And despite the fact it says best prognosis on that slide, which I'll dispute for a couple of reasons. One is the fact that the best prognosis is not to have cancer, and that isn't me being flippant, that is something which I think is always best to bear in mind. But also, it depends how you measure it. If you take this data and you take it longer term, actually recurrence is a real issue. And the particular thing is when we get metastasis, so when the tumor spreads throughout the body, we really are limited in what we can do to improve patient outcome. So what we've been doing for the last few years is looking at the steroid hormone receptor, the estrogen receptor, which drives the growth and the proliferation of these ER-positive breast cancer tumors. And the way these nucleoreceptors work is, in the case of the estrogen receptor, it sits bound by heat shock proteins in the nucleus, and there it basically waits for estrogen to come along, which can diffuse across the plasma membrane and just into the nucleus, where it then causes the ER to shed those heat shock proteins through conformational change, form a dimer, and that binds as a transcription factor to what we call the estrogen response element, but it's a motif, so it's just a standard transcription factor, and it builds these lovely complexes on the gene and the enhancers and the promoters, which then cause transcription to occur. And this is something that over the last 10, 20 years has been methodically worked out. And when I was at my previous position, one of the technologies we helped develop along with the Carroll group in Cambridge was this thing called QPlex Rime, and that's the data I'm showing on the right-hand side of this slide. And what that uses is quantitative proteomics to analyze what's in that nuclear receptor complex. And what you can see here is on the left-hand side, we have basically the estrogen-free complex, and that's where we see these heat shock proteins. And when we add estrogen, we shift to the other side of the volcano plot, because that's what the differential binding is shown here, and we see all these other proteins appear: P300, FOXA1, GATA3, all the things that have been worked out over the last few years, plus plenty more. So we've got a really nice method for finding the parts of our estrogen receptor complex. But we're interested in more than just producing lists, as is everyone. We want to understand how these parts go together, how they function. And what I'm really showing here is what we get from one of those experiments is very like this car or this picture of the night sky or this painting, is the parts themselves aren't particularly useful. I mean, you can go off and do things with them, but what you actually need to do is work out how they work. So you need to know how they come together into whatever it is you're studying. And the way we can then use that to work out what the function is, and this isn't a particularly novel idea, it's what we do all the time, is if you want to work out what a bit of a car does, you would say you want to know what the brake cable does, you'd cut the brake cables. Now, obviously, don't do this on your actual car, but what you'd then find very quickly is the brakes stop working. And that's really the idea behind what we're doing in the lab, this idea of breaking various parts of the ER signaling mechanism to understand how it works. And that's what we were doing for the last few years using shRNAs. So we put these in, target various cofactors we pulled out of the QPlex Rime, and we'll monitor the results by RNA-seq, because every time there's one of those complexes bound to a gene, it's modulating the expression of that gene. And when you collect that data, so this is some of the data we generated in the lab, you can form this matrix. And what you can see here is a standard heat map where it's red, we've got the genes turning on more, and where it's blue, the genes are turning on less. And we can then focus that down on the known estrogen responsive genes. And by analyzing that using one of many data learning learning techniques, this one shown here, it was done using a nested effect model, we build a hierarchy of how the different cofactors we perturbed are regulating that transcriptional response. And the way we interpret what we get out of the network is different genes have different protein complexes on them. And those different protein complexes are really important for us, because what we want to know with the estrogen receptor is which complexes are really involved in actually causing the genes to do proliferations, and which genes got a different complex that perhaps aren't so important. And when we talk about breast cancer and relapse, that's really crucial, because our key target at the moment is the estrogen receptor. And what this would allow us to do is work out what cofactors we could also target alongside the estrogen receptor to help those patients. But this is quite a slow technique. And what we've put forward to sort of build on this method is using the new single cell methods that come along. So this is an example of Perturb-seq, where what you can do is you can combine a Cas9, a barcode, and a guide RNA all in a vector. You make a virus and you put that on your cells. And what this data is showing, and this was done by a PhD student, was analyzed for us, is you then sequence those cells, and you can use that barcode to then group together all the cells that have the same guide RNA. And that's very much like what I showed you with the shRNA. But instead of having to do 3 replicates in 3 Petri dishes, a huge amount of cell culture, you can do it all in 1 Petri dish in one go, and massively increase the throughput. And what the sort of dotty picture on the right showing there, if you haven't seen one of these before, this is one of the dimensional reduction techniques like PCA or UMAP or t-SNE, which you may have come across, and each dot represents a cell. And what's really nice here is the dots are clustering by the guide RNA they receive. So we now have a way of rapidly generating changes in the transcriptional response depending on which parts of that system we've broken. And of course, what we can also do is give them the steroid hormone of interest to see how it changes that response. So we can also rapidly increase the amount of data we're collecting. And this is what we got up and running in the lab, and this is on the estrogen receptor. And our plan now is to take it to the glucocorticoid receptor. And the key reason for that is that this paper from Mohammed recently showed that in triple negative breast cancer, another form of breast cancer, the glucocorticoids, the ligand for the glucocorticoid receptor, cause the GR to promote metastasis. Yet the same compounds actually cause some types of cells to go through apoptosis. And we're really curious to see what the changes in function of those cofactors are, and if we could tweak it so maybe we can prevent that metastasis in breast cancer in the breast cells, but keep the essential anti-inflammatory properties which are needed to treat for patients and all the side effects they have. So, because I realise, because of technical difficulties, I'm a bit behind on time, but we basically apply the same method I've just shown you here, represented in a different way on the GR, and we can see all those cofactors. Many of them are very similar to the estrogen receptor because it's the same family complexes. And our overall aim is really to wrap this all together. And this is a summary slide from a grant I recently was awarded, but focusing on the part 3 there, if we can sort of work out which of these cofactors or assign them key functions in different tissue types, there are plenty of compounds already there to target these cofactors. So our aim, if we're really fortunate and we get all this to work out as we're planning, will be to start to block some of these cofactors and see if we can eliminate that metastatic effect of those steroids used to treat inflammation, but not block those immune cells, which are so important for patient outcome. So I just want to say thank you to the funding I've got. As I say, we've just been awarded BBSLC funding, so I haven't made it onto the slide yet. To the groups who've helped me here. And we're also hiring at the moment. I have a postdoc and an RA position online, which both close on, I think it's the 14th, so a little bit of time. Thank you, and sorry for running over there, but I hope you don't mind given the little difficulties you had at the start. So thank you for listening.
Thank you so much, Andrew. And yes, no, technical difficulties happen. So we're going to have questions at the end, and I'm going to move on to our next speaker this afternoon, who is Dr. Tanya Soliman from the Barts Cancer Institute, Queen Mary's University of London. Dr. Tanya Soliman was awarded her PhD from the University of Queensland, studying the role of MAP kinase signalling in the cell cycle and mitosis with Professor Brian Gabrielli. After completing her PhD, she moved to London to start a postdoctoral fellowship with Professor Peter Parker, initially at the CRUK London Research Institute, and then moving to the Francis Crick Institute when it opened in 2015. Here she investigated the genome protective role of the kinase PKC epsilon in the cell cycle. In 2019, she commenced a postdoc with Dr. Sarah McClelland at the Barts Cancer Institute, researching chromosomal instability in high-grade serous ovarian cancer, and has started her own independent laboratory at the BCI in July 2020. Tanya's presentation today will look at Aurora B kinase, a key regulator of mitosis and the functional consequences of combinatorial activation loop phosphorylation. Over to you, Tanya.
Great, thanks very much, Amy. Sorry, that was a little bit of a mouthful. So thank you very much for that introduction, and also to the Biochemical Society for the opportunity to talk to you about my work today. So I'm going to discuss some of the work that I did during my postdoc at the Crick, and also give you a bit of a flavour as to how I think the new directions that we're going to take now that I have started my own group at Barts Cancer Institute. So just so that we're all on the same page, I thought that we would start with a couple of kind of fundamentals. First of all, what is a kinase, as those are the kind of main enzymes that my lab is going to be focused on. And these are enzymes which catalyze the transfer of phosphate to a serine, threonine, or tyrosine of a substrate protein. And this process of phosphorylation affects every biological process that you could probably think of. So a kinase is made up of a catalytic domain, and this consists of both an N lobe and a C lobe. And sitting in the middle of that is the activation or T loop. And this is quite often phosphorylated, and this facilitates the correct conformation of the kinase to facilitate its catalytic activity. Then between the N lobe and C lobe is the catalytic cleft, and this coordinates nucleotide binding, ATP binding, and also substrate binding. So what happens is the substrate comes in, it meets with the kinase, the gamma phosphate from ATP is transferred to the appropriate serine, threonine, or tyrosine residue, and the substrate can then go off to do its thing. So the effect of phosphorylation differs between substrates, and it can have effects such as localization, changes in activity, influence protein-protein interactions, stability, and so on. But the vast majority of biological effects of individual phosphorylation sites are actually unknown to us. So the second kind of thing that I wanted to make clear was that mitosis is the final step of the cell cycle, and this is where, this is where cells now segregate both the cytosolic contents and also the genetic material so that it can become its two independent but identical daughter cells. So this process must happen with incredibly exquisite fidelity, as I'm sure you can imagine, and this ensures the integrity of the ensuing generations of cells. So throughout the cell cycle and mitosis are checkpoints, and these ensure that this process occurs as error-free as possible. And many of these signaling pathways involved in both the cell cycle and the cell cycle associated checkpoints are regulated by kinases. So as you probably have guessed, the particular mitotic kinase that we're interested in is Aurora B. And you can see from this IF that it localizes to the chromatin during prometaphase and metaphase, before it relocalizes to the central spindle during anaphase, and then the mid-body just prior to cytokinesis. So, as I've said, Aurora B is a key kinase in mitosis, and what I want to emphasize here and throughout is that while we think all of the things that Aurora does are incredibly interesting, we're really using it as a tool and as a model, because what we think we've discovered about this particular kinase and the structure-function relationships, we think it's going to be applicable to many kinases of the kinome. So as I mentioned previously, most kinases are phosphorylated on their activation loop to put them into the conformation required for their full catalytic activity and stability. So for Aurora B, this is threonine 232. So under certain circumstances, Aurora B is also phosphorylated at serine 227. This is also in the activation loop and close to the activating phosphorylation site. So what we hypothesized was that inserting this new negative charge here may change the shape of the activation loop. And so through a series of molecular dynamic simulations in collaboration with Miraz Rahman's group at King's, we proposed the following structures here, with either one phosphorylation site or two phosphorylation sites occupied in the activation loop. So I'll refer to these as either the 1P or 2P Aurora B from now on, but this simply refers to the phosphorylation state of these two sites, not of the whole protein. So you can see that when we overlay these two structures that they're now different, and the substrate peptides shown here in darker shading adopt different binding positions. So we wanted to know whether these conformational changes had any effect on the activity of the kinase. Biochemically, we showed that this change in conformation does not change the ability of the kinase to either use ATP or its stability. We did this by thermal shift assay. Nor did it affect the phosphorylation of a well-described Aurora B substrate, histone H3 serine 10. So H3 serine 10 is phosphorylated at the onset of mitosis and is involved in the initiation of chromatin condensation, and is quite often used as a kind of gold standard or marker for Aurora B activity. So from here, we concluded that there was no effect on the catalytic competence of the kinase. So what we did was we broadened our investigation into other Aurora B substrates, and we screened these substrates. And what we found was that a number could be phosphorylated by either the 1P or the 2P form. However, what we observed was a number of substrates were preferentially phosphorylated by the 2P form when we conducted our first screen. So because of other ongoing work in the lab at that time, we investigated one particular target substrate that we were particularly interested in, and that was topoisomerase 2 alpha. And what we saw that when we arrayed the entire protein of topo, so chopped it up into peptides and then arrayed onto nitrocellulose, then did kinase assays on top. What we saw was this really striking switch in substrate specificity. So when we did a kinase assay with the 1P form of Aurora B, we identified that it phosphorylates this substrate in the C-terminal tail of topoisomerase, which we identified to be threonine 1460. And then when we used the 2P form of Aurora B, it phosphorylated a substrate in the N-terminal region, which we identified as being serine 29. So we had identified PKC epsilon as an upstream kinase for this phosphorylation of Aurora B at serine 227. And previous work that had been done in the Parker lab had identified 2 inflection points where PKC epsilon was critical for the successful transit through mitosis. And we went on to show that Aurora B serine 227 was the direct target of PKC epsilon in affecting these signaling cascades. So the first point was in the timing of the metaphase to anaphase transition. And as I said, we're now able to place Aurora B downstream of PKC epsilon in the signaling pathway. So when we mutate Aurora B serine 227 to a now non-phosphorylatable alanine, this phenocopied many of the previous observations that we'd made with PKC epsilon, either inhibition or depletion. And this included an increase in DNA bridging in anaphase, and also an increase in the number of catenated sister chromatid pairs, indicating that dysregulation of this signaling pathway results in segregation defects. So then when we looked at the final stage of cell division, cytokinesis, we see that Aurora B localizes to the midbody, where it plays a role in signaling and recruitment of the excision machinery required to sever the bridge between the 2 presumptive daughter cells. So here we were also able to place Aurora B downstream of PKC epsilon, as serine 227 phosphorylation of Aurora B was sensitive to PKC epsilon inhibition at the midbody. Again, when we express the non-phosphorylatable mutant of Aurora B, this phenocopied PKC epsilon inhibition and resulted in cells failing to exit from cytokinesis, and we observed this through an increase in the number of binucleated cells present. So to summarize, it wasn't just that this additional phosphorylation induced a conformational change. This switch in substrate repertoire actually had really profound signaling consequences. So while both forms could phosphorylate a number of common substrates, phosphorylation of serine 227 seemed to occur under conditions of pathological stress, which I haven't had time to go into today. But briefly, the presence of persistent catenation resulted in segregation defects and activation of the spindle assembly checkpoint, and also activation of the abscission checkpoint. Both of these required the fine-tuning of Aurora B signaling to enable exit from these particular checkpoints. So if you're interested in the details of the signaling pathways, the work has been published and the citations are down here below, and I'm happy to discuss offline as well. So as I mentioned before, our initial data and our planned experiments going forward involve Aurora B kinase, but we think that this mode of regulation could actually be common to other kinases also. Almost certainly the other members of the Aurora kinase family are regulated in this way, as they're highly conserved in this activation loop region. And we think that other mitotic kinases may also undergo this form of regulation. For example, PLK1 and 4 can also be phosphorylated on additional sites in the activation loop, while MPS1, which is currently showing promise in clinical trials, is reportedly mutated at a number of residues in some cancer types. So we're also beginning to look at other kinases in other kinase families, such as PKN2 and PAK4, which can also carry these additional activation loop phosphorylations. So when we looked for what we're calling combinatorial activation loop phosphorylation across the kinome by mining the phosphosite database, we found that up to half of the kinome can be multiply phosphorylated on the loop. So some of these are of course more than one additional phosphorylation site, which is going to be difficult to untangle. Nonetheless, the strategy that we're going to adopt is to model these kinases by molecular dynamic simulation, similar to what we did with Aurora B, looking for other kinases which undergo conformational change accompanying activation loop phosphorylation. So the aim of this work is to reveal new and previously undefined signaling nodes, and to determine whether any of these play a role in kinase signaling network rewiring in cancer. So the idea is that by establishing this workflow initially with Aurora B, we'll then have a pipeline by which to examine the effect of combinatorial activation loop phosphorylation in other kinases. And the kind of overarching theme for the lab is to build on our knowledge and understanding of kinase biology and behavior in terms of these structural and functional regulations and how they then become dysregulated in cancer. So that just leads me to acknowledge that this work was a huge collaborative effort by my colleagues in the Parker Lab. And I'm also grateful to our collaborators at King's and Oxford, and also to the BCI and Barts charity for funding going forward. So thank you very much for your attention, and I'm happy to answer questions in the session later.
Thank you so much, Tanya. And as Tanya says, we'll be having questions at the end of the session, so do submit your questions in the relevant box. We'll be moving on to our next speaker today, which is Dr. Dave Boucher from University of York. Dave did his PhD in pharmacology at the University of Sherbrooke, Canada, on biomedical characterization of apoptotic caspase specificity. He then moved to the lab of Professor Kate Schroeder at the University of Queensland to study the inflammasomes as a postdoctoral fellow before joining Professor Peter Brott's lab at the University of Lausanne for a second postdoc in inflammasomes. Since 2020, he has been a lecturer in biomedical science at the University of York, where he leads a research group focusing on inflammasomes and protease signaling. Today, Dave will be presenting his research on detecting lipopolysaccharides in the cytoplasm of immune cells by the non-canonical inflammasome. Over to you, Dave.
Thank you very much, Amy, for your kind introduction, and thank you very much for the Biochemical Society for the invitation. I'm delighted today to present some of the work I did during my last postdoc in the lab of Peter Brott's, and to share with you why I think LPS recognition inside cells is a really cool pathway. So over the years, I've been very interested to understand how pathogens are recognized in cells during infection. It can be pathogens that infect epithelium, like in the guts, or pathogens that infect immune cells, like macrophages. And lately, I've been mostly focusing on understanding how specific ligands derived from pathogen are recognized. And we've been working a lot with LPS, or lipopolysaccharide, which is a component of the outer membrane of gram-negative bacteria like Salmonella, E. coli, or Shigella. And this is truly a very fascinating molecule. So you can see here a picture. So LPS is composed of 3 different parts. O antigen would face the outside of the bacteria and will access the cytosol. Core saccharide, which is in the middle, and a very hydrophobic part called lipidae, which is located and embedded inside the membrane of the bacteria. So our knowledge of how LPS is recognized in immune cells is actually quite well established for many years. So we know, for example, that when bacteria are outside the cells, LPS is sensed by different molecules like LBP and CD14 to be subsequently transferred on plasma receptor called TLR4. And once it's bind to TLR4, LPS would induce immune signaling, which will trigger release of specific cytokine. However, many pathogens actually have a lifestyle where they will actually be engulfed and live inside immune cells and epithelial cells, and we don't really know how they are recognized inside those cells. And so a few years ago, a very interesting paper was published in Nature where they identified a new complex that was responsible for the recognition of LPS inside cells, and this complex was called non-canonical inflammasome, or NCI. So we'll call it NCI for this talk because it's going to be shorter. So the NCI is a complex that can activate inflammatory proteases called caspases, in this case, caspase 4. So when caspase 4 is activated, caspase 4 will cleave a protein called gasdermin D and induce the form of cell death called pyroptosis. However, people believe at the moment that LPS was directly recognized by the caspase, but we didn't believe it was the case because we didn't think that LPS was able to access the lipidae part, which was suggested to be binding on the caspase 4. So to challenge this, we adopted a very simple approach. So we use HeLa cells, which are a model of human epithelial cells, and those cells express caspase 4 on a basal level. And what we did, we left the cells either naive, untreated, or transfected them with interferon gamma. And then we transfected them with LPS to mimic bacterial infection and delivery of LPS inside the cells. And at the end, we measure cell death as a readout of non-canonical inflammasome activation. And what we found was actually quite interesting. We found that only in cells that were primed with interferon gamma, the non-canonical inflammasome was activated in response to LPS transfection. This was suggesting that there was an interferon-inducible factor that was responsible for the activation of this complex, and that caspase-4 alone was not sufficient for that. We saw the same thing if we were doing electroporation of LPS or simply infection with gram-negative bacteria like Salmonella. So to cut a long story short, we found this protein, this family of protein called guanylate binding protein, as being responsible for this phenomenon. So GBPs are interferon-inducible genes, and we have 7 in human, and they all have a dynamic like GTPase structure. So they're composed of the GTPase domain and a bundle domain. And upon hydrolysis of GTP, they will form oligomers. So we first ask what happened to GBP location during a Salmonella infection. And here again, we went back with our ELA cells. So we took ELA cells that were stably expressing EGFP fusion of GBP1 to 7, and we infected with Salmonella in red here. And as you can notice here, in cells primed already with interferon, we see recruitment of GBP1, 2, 3, and 4 to the bacteria, whereas GBP5 to 7 were not recruited. Interestingly though, in naive cells, so cells that were not primed with interferon gamma, only GBP1 was recruited, again telling us that GBP1 was probably the upstream GBPs in the pathway. So that was very interesting to us. So we went and generate knockout lines where we removed GBP1 in ELA cells. And again, we monitored the effect of GBP1 deletion, either with Salmonella infection or LPS transfection. And what we found was quite striking. GBP1 deletion was completely blocking cell death in response to intracellular LPS, suggesting that it was the most upstream component of the non-canonical pathway. We also confirmed that in other cell type, so we use, for example, HBEC cells, which are bronchial epithelial cells, and confirmed again that the non-canonical inflammasome required GBP1 as well as caspase-4 and Gas-WD. We also found the same thing in ACAT cells, which are keratinocyte cell line. So we're very excited about that, and we really wanted to see what were the minimal component of this inflammasome complex. So to do so, we went back to our ELA cells, and we tried to reconstitute the complex and see what were the minimal components required for caspase-4 activation. So what we did, we took naive cells, and we co-expressed different GBP combination and looked at caspase-4 activation in response to LPS transfection. And we're very happy to see that when we co-express GBP1 with GBP3 and GBP4, we can actually reconstitute the cell death pathway in naive cells, again suggesting that GBP1, 3, and 4 are the minimal component required for the activation of caspase-4. So I told you at the beginning that we were looking for a sensor or a way that LPS is sensed, and here we still don't have our answer. So we know that caspase-4 is not the first sensor of LPS in the pathway, but we still don't know which one it is. So we went out and identified actually GBP1 as being this sensor in the pathway. So here we use size exclusion chromatography approach to see if GBP1 can form a complex with LPS. As you can see here, so GBP1 alone will elute as a monomer of about 70 kilodalton on the column. However, if you mix GBP1 with LPS, you will see that GBP1 will shift and form a nice protein complex, suggesting that indeed the interaction is direct. Interestingly, we found that GBP1 was not involved to interact with lipid A, suggesting that indeed it was interacting with the other part of the LPS that is accessible to GBP1 in the cell. We confirmed this using surface plasmon resonance and found a good interaction between GBP1 and LPS of about 64 nanomole. We also found that incubation of GBP1 with LPS was strongly increasing the GTPase activity of GBP1. Again, supporting the idea that LPS can directly activate and promote oligomerization of the bacteria, the GBP1 on the bacteria. So we came out with this very simple model. We have a lovely paper that's been published last year in Nature Communication that I highly recommend you to read, where we found that cytoplasmic bacteria, after they evade the vacuole, will get detected by GBP1, and GBP1 will recognize directly the LPS at the surface of the bacteria. So we used different approaches to verify this, and we're quite convinced that GBP1 is really interacting with LPS at the surface. So once GBP1 is recruited, we know that GBP1 can recruit GBP2, 3, and 4 and form a nice coat around the bacteria, and then subsequently recruit caspase-4, which get activated on the surface of the bacteria. So we believe that the bacteria will become a signaling hub that will allow cell death upon cleavage of Cas-Vitamin D. So I just started my group. We just have a baby group, but my baby group is interested to understand what's happening downstream of this pathway, and we're characterizing different substrates of caspase-4 in this pathway and studying how this inflammasome is also assembled on the bacteria. This work was performed in the lab of Petrobras, also with Jose Santos, who was a postdoc at the time, who helped me a lot. Yeah, that's all for me. Thank you very much.
Thank you, Dave. Really nice talk. And again, if you have questions for Dave, please do them in the chat. And we're moving on to our next speaker, which is Dr. Tan Wen from Monash University, Australia, who is talking to us live from Australia and stayed up to be with us. I believe it's around 2 a.m. where you are. So thank you very much. Dr. Tan moved to Melbourne, Australia from Vietnam to undertake his bachelor and PhD at La Trobe University. He completed his PhD in 2016 in the lab of Professor Mike Ryan, investigating the regulation of programmed cell death mediated by mitochondria. As part of his thesis, Tan published his first author paper in cell death and differentiation. Since the completion of his PhD, Tan has been working as a research fellow in Dr. Michael Lazarou's laboratory at Monash University, focusing on mechanisms of mitophagy driven by the Parkinson disease proteins PINK1 and Parkin. Tan has made important discoveries into the functions of ATG8 during PINK1 Parkin mitophagy, changing the way that was previously thought about the functions of these proteins in selective autophagy. Today, Tan will present his most recent work on the ATG4 family of cytosine proteases, which has revealed exciting and unexpected roles for the ATG4 family in regulating mitophagy. Over to you, Tan.
Thank you, Amy, for the introduction, and I'd like to thank the organizer for the opportunity to present my work here. Now today I'll be talking about the functions of ATG4 family proteins during PINK1 and Parkin mitophagy. So PINK1 and Parkin are mutated in Parkinson's disease, and this disease is characterized by the loss of dopaminergic neurons leading to motor symptoms. Now, mitochondrial dysfunction has been shown to be a key player in this pathogenesis of this disease. Most PD cases are late onset, however, 15% of them are early onset due to genetic changes in several factors. And among these factors, mutations in PINK1 and Parkin are a common cause of Parkinson's disease. Therefore, study their function can provide insights about the disease mechanisms. So let's have a look at the PINK1 and Parkin mitophagy pathway. Now, mitophagy is a selective type of autophagy to remove damaged mitochondria through lysosomal pathway. When mitochondria become dysfunctional, PINK1 accumulates on the surface to recruit cytosolic Parkin to mitochondria. Parkin is an E3 ligase protein that builds ubiquitin chains on the surface of mitochondria. And these ubiquitin chains are then responsible to recruit the autophagy machinery to build a double membrane structure called an autophagosome to ultimately engulf the damaged mitochondrion. Now, the complete autophagosome then fuses with lysosome for degradation of its content. So central to the Pink1 and Parkin mitophagy pathway autophagosome biogenesis is this ATG8 family protein of ubiquitin-like proteins with LC3 subfamily and GABRAF subfamily and 3 members of each subfamily. So previously, ATG8s are thought to be essential for cargo sequestration during selective autophagy, meaning that without ATG8s, mitochondria cannot be sequestered within autophagosomes. However, in 2016, when I knocked out all of these ATG8 proteins to create a hexa knockout cell lines, what I found was that ATG8 are not essential for autophagosome biogenesis. So in the hexa knockout, mixing all the ATG8s, you can see that mitophagosome or mitochondria containing autophagosome still formed, even though these structures are smaller than the wild type. 3D reconstruction of these EM images from the hexa knockout showing that fully sealed autophagosome were present, as you can see in green here, and mitochondria in red were correctly engulfed within these autophagosomes. And we also showed that ATG8s are crucial for autophagosome lysosome fusion. So after I finished this project, I decided to work on something that related to ATG8s and to stick to my gene knockout expertise. So I chose the ATG4 family of cysteine proteases with 4 members in humans. Now, these proteins are thought to be function during autophagy through ATG8s by cleaving ATG8 precursors to allow these proteins to be attached to phosphatidylethanamine or PE on autophagosomal surface, or autophagosomal membranes. And also yeast studies and in vitro studies suggesting that ATG4s can also remove ATG8s from PE on autolysosome to recycle these proteins. However, the contributions of ATG4 to mitophagy remains unclear. So I decided to knock out this family in HeLa cells using CRISPR-Cas9 to create a ATG4 tetra knockout, as you can see here. Now, I induced mitophagy by using antimycin A and oligomycin, which are mitochondrial toxin to depolarize mitochondria. So in wild-type cells with Parkin expression, you can see that upon mitophagy induction, COX-2, a mitochondria matrix protein, is degraded, indicating that the mitophagy is active. However, in the absence of ATG8 or ATG4, you can see that COX-2 is no longer degraded as quantified here. This indicates that ATG4s are required for Pink1 and Parkin mitophagy. So I went on and looked at mitophagosome formation in these cell lines. As expected, the ATG8 hexa knockout missing ATG8s, the mitophagosomes are smaller than the wild type. Excuse me. So if ATG4s function only through ATG8s, then you would expect that the ATG4 tetra knockout should have similar phenotype. However, the ATG4 tetra knockout did not show any complete autophagosomes. Instead, you see these autophagosomal precursors called phagophores coupling mitochondria. So this suggesting that ATG4 either in ATG4 tetra knockout, the unprocessed ATG8s are inhibitory, or knocking out ATG4s in the hexa knockout, or ATG4s has an ATG8 independent function in this setting. So I therefore decided to clarify these possibilities by knocking out the ATG4s on top of the ATG8 hexa knockout to create a ATG4 ATG8 deca knockout. And when I did that, I saw similar phenotype with the ATG4 tetra knockout in the deca knockout. So I saw no mitophagosomes. Instead, I saw phagophores on mitochondria, and it can be quantified here. So these results indicates that ATG4s can regulate phagophore progression independently of ATG8s. And I was be able to rescue the ATG4 ATG8 deca knockout phenotype by expressing wild-type ATG4s as well as protease-dead ATG4s. Supporting that ATG8 independent function of ATG4s does not require the protease activity. So the question then is, how do ATG4s promote autophagosome biogenesis? The challenge with these proteins is that we do not see these proteins associated with mitochondria during mitophagy. So I decided to use proximity biotinylation using APEX2 approach in collaboration with Christian Behrens in Germany, and using mass spec to study the mechanism of these proteins. So with this approach, we identify the proximal interactors of ATG4s, including the proteins involved in transport and membrane trafficking, also proteins associated with lipid, and also proteins that are previously linked to autophagosome formations and ATG9 vesicles. So the proximity interactome points toward a role for ATG4s during ATG9 trafficking in nucleation. So therefore, I went on and looked at the mitochondrial recruitment of ATG9 during mitophagy in the hexa knockout versus deca knockout. So in the hexa knockout, you can see here in green is ATG9 coming to mitochondria on the surface, whereas when you knock out ATG4s, this recruitment is abolished as quantified here. So ATG4s regulate ATG9 trafficking during mitophagy. And recently, ATG9 and ATG2 have been shown to function together to transfer lipid from ER to support Factor 4 growth. So what I decided to do is to use ATG2B as the marker for Factor 4, and we using correlative light EM and focus ion beam scanning EM and coupled to machine learning to be able to visualize this GFP ATG2B structure in the hexa knockout versus the deca knockout to see what's going on in this cell line. And this work was done by a postdoc, Ben Padman, in the lab. So we first located the GFP ATG2B with confocal imaging, and then subjected the samples to clamphysem. And then we collect a large data set. We also developed an AI-directed approach to be able to reconstruct this large volume of the cells, and then zoomed into the phagophores, as you can see here. So in these, you can see the examples of phagophores from hexa knockout and the deca knockout. Phagophore in green, mitochondria in red, and ER in blue. So let's have a look at a closer look to the phagophore in these cell lines. So in the hexa knockout, you can see green is phagophore cupping mitochondria in close proximity to ER in blue. Now, in hexa knockout, you can see that these phagophores are bigger, and also sometimes they're poking into the ER membrane and in close a lot of ER surrounding the phagophore as well. However, when we knock out ATG4s, you see that these structures are smaller and the ER membrane is less. And we will be able to quantify this to show that in the absence of ATG4, you see that phagophore volume as well as phagophore ER contacts also affected. So it's all the data suggesting that ATG4s regulate ER phagophore contacts via ATG9 trafficking to stimulate phagophore progression independently of the protease activity and ATG8s. Now, this is a working model here. So in this model, ATG4s can function through ATG8s to support phagophore growth, but also can directly regulate trafficking of ATG9 to the phagophore via ATG9 or via the proximity interactors. And one of these interactors we identify is LRBA, a protein mutated in autoimmune disease. So it is really interesting to see whether or not ATG9 trafficking as well as mitophagy has any role in these disease models. So with that, I would like to thank the people involved in the work, and again, thank you, the organizer, for the opportunity, and thank you for the listening. Thank you very much.
Thank you very much, Tan. And so Tan was our last speaker today. So we're going to move on to our Q&A session. So I'd like to remind the audience to send in their questions stating who the question is for. We've had some come in during the talks already, but it would be nice to see a few more. So I'm going to start with a question for Dr. Tanya Soliman. So one of our audience has asked, do the other members of the Aurora family have the same substrate specificity as Aurora B? Are you able to take that?
Sorry, I missed just the last bit of the question. Would you mind repeating it for me?
Sure. The question is, do the other members of the Aurora family have the same substrate specificity as Aurora B?
They have the same recognition motif, and there is a paper which describes that Aurora A is also phosphorylated on the same or homologous site to Aurora B at serine 227. So the authors of that paper actually say that it induces a loss of function, but at the moment, one of the projects that my new PhD student is doing is looking to see where actually that is a change of function, which is what we kind of suspect that it might be.
Okay, thank you. While we've got you speaking, I'll give you another question from our audience member. So these data also suggest, your data also suggest a critical role for phosphatases in regulating signaling outcome. How will you incorporate phosphatases into your approach, and which phosphatases are implicated in regulation of Aurora B function? Is this a major mechanism for phosphatases to regulate signaling output?
Yeah, so we haven't got any plans, to be honest, with the phosphatases yet, but it is something that kind of always sits in the back of my mind as to what the kind of, you know, yin and yang is, I guess, with phosphorylation. So one of the phosphatases known to dephosphorylate Aurora B is PP1, and so that's something that we're going to keep in mind moving forward.
Okay, thank you. Our next question is for Dave, and they ask, has a similar intracellular innate immune response been found against viruses or viral features in humans?
That is a very good question. So we know that GBPs, for example, can be important for recognition of viruses, but as far as we know, they don't trigger the activation of the same pathway. However, we know that viruses can be recognized through other inflammasome complex. So there's other innate immune sensor that can also recognize virus, and they can trigger a similar response, but this time activate another protease that is called caspase-1.
Thank you. I'm just scrolling through the list of questions that we have. So the next one is for Andrew. You've spoken about increasing the specificity after targeting the glucocorticoid receptor to avoid exacerbating inflammation, but how can you target it to a particular cell type?
I mean, this is basically the key question of the research. It's a great one. We don't believe all cell types are expressing exactly the same cofactors, so it's all about finding those networks, and we're going to be looking at multiple tissues. So we're looking at T cells, breast cells, and breast epithelial cells, and bladder cells, because it's got very different characteristics. And if we can isolate cofactors that either do a very different job or have a very different expression pattern in those tissues, they're the ones we really are going to sort of follow up on. And there's obviously going to also be an epigenetic component there, so how the landscape is. So it might be that the cofactor is in multiple tissues, but its function is less important in one than another. So if we start inhibiting that, we can get the precise response we want without inhibiting another response in another cell type. And obviously, because the immune system invades tissues by its very nature, it's really about that immune versus the local tissue that we're going to be looking for the comparison on.
Thank you. So we've just had another question in for Dave. LPS binding to immune cells is explained here by Dave. Does the binding of LPS to tissue cell follow the same pattern?
Yeah, so we believe that there's a bit of difference there. So for example, we believe that TLR4 is recognizing the very hydrophobic part of LPS, so the lipid A part. But we know in the case of GPPs, it really recognizes some charges that are actually located inside the sugar core of the LPS. So it's slightly different.
Thank you. And for Tan, how does the novel role of ATG4 family link into human disease and it's the role of autophagy?
At the moment, obviously, we're not sure. It's a great question. There's no mutations yet identified in ATG4s to be involved in human disease. So yeah, I would love to have something like that, but yeah, we haven't found any mutations in ATG4s.
Thank you. Another question for Dave. Is the GTPase activity of GP1 required for its function? Which domains of GBP are responsible for their heterooligomerization?
That's an excellent question. Yes, so we need the GTPase activity for the oligomerization and the formation of the complex on the bacteria. However, the binding to the LPS does not require directly GTPase function. And so we know that the GTPase domain can allow dimerization of the GPPs, but that the bundle domain, the last bit, is also important for multimerization of the GPPs.
Thank you. And to wrap up this question and answer session, we have a general question to all of our speakers today. What tips would you give to fellow early career researchers in terms of pursuing a postdoc or starting their own laboratory?
I would like to start by saying that if you can have like a good mentor to support you throughout the process, that could be great, because then you know, everything will be easier. You can get experience from them, you get advice, and for example, my supervisor is really supportive of what I'm doing and trying to direct me through the process. So that was great for me. Yeah.
Thank you. So we will begin the wrap up this session. I'd like to thank all our speakers once again. Let's continue the conversation online, and you can follow at BiochemSoc and at PPP Publishing on Twitter and tag them in your comments about talks you've heard today. I'd also like to spotlight a related event, the 88th Harden Conference on Kinases and Pseudokinases. The conference will take place in 2022 and will be a great opportunity for those working in the field of kinase signaling to gather and discuss the latest research. But as the event has been postponed from this year, which is the 30th anniversary of the publication of the first protein kinase structure, an online symposium on the topic will be hosted later in 2021. Keep an eye on the Society's website for details of that. If you have an idea of a topic to be included in the webinar series, please do let us know. You can submit a webinar proposal form on our website. Visit www.biochemistry.org/webinars to submit your topic idea or to view previous webinar recordings and see what's coming up. Next time in the Biochemistry Focus webinar series, we welcome Professor Antoinette Gillian and Tim Daffron to present on amphiphilic polymers for membrane protein solubilization and characterization. That will be on the 25th of March. Register for free on our website now. Lastly, I want to highlight that it's more important than ever to stay connected. Joining the Biochemical Society community of researchers and specialists is a great way to keep in touch with your fellow molecular bioscientists, and members can take advantage of a variety of benefits such as discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, and personal online access to 2 journals. Visit biochemistry.org to find out more. So thank you all and goodbye.
Thank you.